Welcome to this first session of the OmicsLogic Data Science program. In today's session, our team will be sharing information, examples, and an overview of OmicsLogic training resources designed for anyone to learn data science for biomedical data, as seen in application to projects sourced from current topics in biomedical research. In this 1-month program, we will combine online sessions like we're having today, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in our well-known and respected OmicsLogic programs. To begin with, let me introduce you to the resources that we will be utilizing in this program. The first resource is our educational platform. I am putting the link to the educational platform in the chat box. In this platform, you will find introductory courses to projects, which will serve as a step-by-step guide and will help you as a springboard into the world of bioinformatics. Our next platform is a powerful tool to help anyone perform NGS data analysis. We have created a platform that has over 30 different pipelines and enables anyone with a computer and an internet connection to perform complex bioinformatics analysis by running bioinformatics pipelines in the cloud. And I will be giving you an overview of these platforms very shortly. Our third platform is the Code OmicsLogic, which offers practical coding assignments to help you understand the R and Python code elements that will be part of any data science project you end up working on in the future. We have a question here from Ashtin. And Ashtin, we will be answering your question very, very shortly. And Ilya will be addressing them in a while. But before we can answer your questions, let me take you to the different resources and show you the registration process for the program, as I just realized that there are a lot of users who have joined us today for the first time and may not know how to register for the data science program. So the EduDBioinfo platform that you see right here, and I've also shared the link again in the chat box, is the first platform, as I explained, which will help you get acquainted with the different multi-omics courses. So if you see here under the courses, there are different courses which are covered. One of the main courses that we suggest that everyone take is the Introduction to Bioinformatics. So this is a free course, and if you are just joining our platform for the first time, we definitely recommend you to go ahead and take this course. And if you have already taken this course, then you know that what this course talks about. It's a very basic and introduces you to bioinformatics, and this will serve as the starting point for the program. And later on, we will go on and explore more courses. Now, coming to the program registration, a lot of you may have already seen this program registration page. And in this page, you will have all the details of the different sessions of the program, what the program is about. As you see today, we are here on Introduction to Data Types and Properties that Ilya will be talking about very shortly once I finish this overview. And you must have registered via this form here, right at the bottom of this page. Now, if you have not already registered and are interested in registering, you can still register via the link here. And I will also send out the links in case that you are not able to access the chat right now or are watching from your mobile phone and cannot register. So I will send out an email to everybody after the session along with the video recording that you can access the links easily. So once you're registered for the program, to complete your registration, you need to just go on this page. It's called datascience.omicslogic.com. And we have several different options here to get registered for the program. And very quickly, I'll give you an overview of what the beginner, intermediate, and advanced options mean. The beginner option is perfect for anybody who is just getting started in your data science journey and wants to learn the program. This will give you access to the full program and all of its resources. While the intermediate program goes a step ahead and gives you access to a mentor who would be available to work with you on a one-on-one session. So in the beginner program, we will have group meetings like we're having today with everybody. But in the intermediate membership option, you can also request for a one-on-one meeting with a mentor who can solve any questions that you may have. While the advanced option gives you an additional 30 days to work on your bioinformatics data science project, and also collaborate with our research interns who are part of our community and work with the mentors on developing the project. So this is recommended for anybody who is sure that they would want to develop a project and needs that amount of time, which is beyond the next 30 days, to work and develop this project. So once you are registered here, you will be finding all the payment details. And if you have a coupon code, you can add that coupon code and register for the program. Abdur has a question, why is this program not free? So Abdur, the resources that you see, as well as the time of the mentor who will be assisting you during these sessions, cannot be made free at this point of time. We do hope that we are able to offer this to the maximum number of people, and hence we're having a free session like this one today. So thank you again for joining. And once you are registered for the program, you will receive an email from my end, which is marketing@pine.bio. So this will enroll you into this organization page, which is called the Omix Data Science Organization page. Now the organization page is going to be like a classroom where you can have an update of all the sessions and all the events that are there within this particular program for the next 30 days. So here you see, today we are doing the session 1. The next session is going to be on 14th January. So the program is a mix of self-learning as well as online sessions. And the way we mix it up is you will be given access to asynchronous courses. Let me pull it up here. So these asynchronous courses, you can take them at any point of time during this 1 month period, and they cover different multi-omics topics and are related to the program. What we will actually do in the sessions is go beyond the courses. Like we will take sometimes, for example, we'll take up a part of this course and explain them during the sessions, explain the logic of analysis, help you run the pipelines together and explain what these pipelines mean. And once you are familiar with it, you can go ahead and do more courses. And the reason this exercise has been designed is to help you understand first and then apply the same logic to your own bioinformatics problems or biological questions. And as you see in this organization page, you also have a forum. So for example, in the forum here, we have also given you a tutorial on how to get started with installing R and Python, which is going to be a feature in this program. We are going to be covering a lot of this. And then moving ahead, one thing that all of you must do, even if you're registering for free on the EduT Bioinfo platform, is update your profile. Right. So yes, there are quite a few medical doctors who take our courses, and we will be getting to this question very, very soon once I show you the profiles here. So here, as you see, this is a complete profile of an individual. And once you have completed your profile and you have attended one of our programs, we actually give out these certificates and we will add them to your profile. So your profile can serve as a CV or as a way for you to display this or share this with any of your peers and help them get to know you better or know where your skill sets are right now. As you see here, Simai has also done an independent project of her own. So that is also added to your profile. So this is why you need to update your profile, have a full name, add a profile photo, and complete your profile. Again, for researchers especially, you can also add your ORCID IDs, you can add your LinkedIn details as well as your Facebook details or any other social media that you would be intending to add. Now, once all of this is over, the second platform which we will be utilizing a lot to run the pipelines is the T-Bioinfo platform. As you see, I'm signed in here successfully. So to just give you an overview, if you click on one of these, I'm just showing you a demo pipeline, you can see that here is a full set of an RNA-seq pipeline. So like this, we will be going through several different pipelines with your mentors, and Ilya today would be giving you an overview of this. Okay, Najia, so I will be showing you how to create a profile right at the end and getting to the questions very, very soon. And now, as I was talking about the pipeline, so this is the platform which you will have access to. You cannot create access on the server T-Bioinfo platform, but once you register for the program, you will be getting access to this platform along with 5 TB of space in the cloud to help you run the pipelines, perform analysis, and learn from it. And the third platform, as I have shown you earlier, is the OmicsLogic Code Playground. Here you have different courses on getting started with R and Python. This is again a platform which you can register for free. I have put the link to the platform in the chat box again. And we will also be utilizing this platform extensively to learn from the different resources. Now, before I go back to sharing the slides, as I see there are a lot of questions here in the chat box. We will be answering them right at the end of the presentation so that we can effectively answer everybody's question. I would also invite you to join our communities in LinkedIn. ATN, could you kindly check your internet connection? And Aditya, if you're able to help ATN with the audio issue, you need to join via Zoom audio. And also, sorry again, and also invite you to our Facebook groups. So via these groups, we reach out to our wider community and you are able to interact with everyone else who are part of this network. Now, let me get back quickly to the presentation and then pass it on to Ilya. Okay, so now I would like to introduce you to what Pine Biotech does. So what we have been discussing here today is a result of hard work by an amazing team led by company co-founders, Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team you see here are experts in bioinformatics, computer science, and training, and some of them will be presenting today as well. The OmicsLogic Data Science program is designed to offer an introduction to statistical analysis and machine learning, and a broad overview of technologies and project examples to learn about, and develop practical data science skills using user-friendly cloud technologies as well as R and Python coding. After completing the program, you will gain experience with methods for analysis of big data and data wrangling, essential skills for bioinformaticians as well as those interested in other types of non-biology related research using data analysis. The program is designed for students starting from the undergraduate level to faculty and researchers from both biology and clinician backgrounds who can participate to learn more about data science in biomedical research. In the upcoming OmicsLogic program, you will have the opportunity to interact and collaborate with an international team of experts specialized in the application of data science to biological data sets. Let me introduce you to the program mentors. Dr. Harpreet Kaur is our certified OmicsLogic trainer and expert bioinformatician who will guide the hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is cancer genomics and machine learning. Dr. Mohit Mazumdar is our OmicsLogic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. Ilya Brodsky is the Biotech co-founder and CEO. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. I will now invite Ilya to speak about the increasing significance of data science in biomedical research and introduce you all to the types of challenges we will be addressing in this upcoming program.
Thank you, Bibsa, and thank you everyone for joining today. As there are many questions in the chat box, I wanted to just ask everybody to hold on with the questions until we are done. And the reason for that is because I think many of the questions will be addressed in today's presentation. So before we start, I also wanted to ask everyone here a question, and I'm going to launch a poll where there are 3 questions. So the first question is, what country are you from? And we only have a few options here, but if you're any other, then please write which country you're from in the chat box. The second question is, what is your level of education? High school, bachelor's degree, master's degree, PhD, or other? And what is your area of interest? And here you can select multiple things: bioinformatics, biotechnology, cell and molecular biology, computer science, data science, infectious diseases, oncology, or others. Okay, and actually, yes, I see there's a 4th question. So what are you expecting to get from this program? Again, you can select multiple things here. And by the way, just to let you know, this data is also going to be one of our first data sets that we will work with in our data science training. So we'll see how to work with a data set that comes from something like a chat box, try to analyze it, understand what are some major trends in all of the programs that we run. So thank you for filling out this survey. And we will keep this up just for a couple more minutes for everybody to have a chance to answer. So we have about half of you have answered already. So if you have not yet, please complete the survey. And again, if you are from another country that is not listed here, you can put out your answer, write it out in the chat box. Okay, so let's take a look. Okay, I see Canada, Pakistan, US. Okay, there is a US option in the poll, so please use that also. Bolivia, Honduras, UK. Okay, thank you, Pooja. Italy, Pakistan, Malaysia, Brazil, Japan, Israel, Azerbaijan, South Africa, India, Finland, Argentina. Okay, excellent. So looks like we cover the whole world. Korea. Okay, excellent. Thank you everyone for sharing that information. Saudi Arabia. Okay, excellent. Thank you. Okay, so it looks like most of you have answered. Ethiopia, Pakistan, okay, great. We will end the poll for now. Mexico, great, thank you very much. So please complete in just one more minute and we will end the poll. Because what we want to talk about is some basic practical skills. How do we load data? How do we analyze the data? What is data science all about? And how does it relate to biomedical data, which this program is going to focus on? Okay, excellent, thank you. Japan, Kenya, Peru, Mexico, excellent, thank you very much. So let's end this poll and we will share the results of this poll in just a little bit. And in the meantime, let's go on to talk about data science. Okay, so this program is going to be focused on biomedical data. It's not just going to be data science about anything, it's going to be specialized on biomedical data. And because of that, let's just think about what is the general idea behind data science. So data science is the overlap between data or information technologies, which deals with data storage, organization, retrieval, data formats, different ways to manipulate that data, statistical analysis or mathematical analysis of data and being able to work with that data, extract meaningful insights, hypothesis testing, etc., and some kind of a specialization domain. And so when we talk about biological data, which is just one of the types of data that you could specialize in when we talk about data science, we end up having a lot of additional information and considerations that come from biological research or the applications of data science to understand some particular type of an experiment or data type, for example, clinical data like the clinical trial data, efficacy of different drugs, etc. So there is a heavy focus on the type of data when we talk about data science in general. When you join programs on data science, they tend to be data agnostic. But this program is specifically for biomedical data. And that means that we will use examples from biologically relevant sources, which we will describe in actually our next session. But today, let's talk about what has happened in general with this type of data over the past several decades. Since the time that the human genome was sequenced and assembled, there has been an explosion of this kind of data, omics data, that has changed the way that we understand different diseases and different types of biological phenomena. That has been driven by high throughput sequencing or high throughput data in general. It's the ability to automate data generation. We can see a clear example of that in the current pandemic. For example, in the past 5 pandemics, the current pandemic has been the one that has accelerated the pace of generating real-time or close to real-time next-generation sequencing genomic data for viral genomes at a tremendous speed. So you can see here a comparison that we did in the May of 2020. And we see here about a 5-fold increase for the number of data sets that are generated per month about the novel coronavirus. Now, this seems to us today as almost trivial. We talk about different types of mutations, we talk about the rate of infection, we talk about all of these data types that previously have not been as prominent and as easily understood. So the amount of data, the number of data sets coming in per period of time, the variety of this type of data is changing. And because of that, data science now is not just for data scientists. Data science is for anybody that deals with biomedical research or that has an interest in biological research in general. In fact, in biological papers, in the publications, there are less and less publications that do not deal with a large quantity of data that has to be processed and analyzed. And so the importance of data science skills is growing in this domain. So one of the major types of data that we will be talking about are omics data types. So what are omics data types? Omics data types are data generated through high throughput automated technologies for example, next generation sequencing, but not only. For example, one of the first data types is Phenomics. Phenomics talks about external characteristics of a sample. That could be a tissue type, that could be cell morphology, it could be description of what that sample is. And as we can generate a lot of this data, it also needs to be processed and analyzed. But the next type of data that probably many of you are familiar with is genomics. Genomics means variation at the DNA level. So it has to do with single nucleotide variation or multinucleotide variations like copy number variation and other variation in the DNA code. Another type of data is called epigenomic data. So epigenomic data is data that can characterize epigenetic regulation. It can include whole genome bisulfite sequencing data that helps us study DNA methylation, or it can help us characterize histone modifications that help the chromatin to be contracted or expanded, more accessible or less accessible, and interact in different ways with other parts of the chromatin. We can also talk about the product of epigenetic regulation, which is transcriptomic data, or data that speaks about gene and transcript expression, so levels of expression and alternative splicing. So these are multi-omics data sets, or data sets that we will refer to in general as omics. Now let's come to the part of the data science. What can we do with such data? So one important part that you need to understand about these data sets is that they speak about molecular detail at the subcellular level, and the data that is being generated can be used to answer certain types of questions. So an important part here is to understand, how is this data generated? Why is it generated? What kinds of questions could be asked of this data? And this is actually a very critical part of a data scientist's job. Many people that come to a data science problem, they come to that problem as engineers. And engineers have a very clear set of rules about inputs and outputs. As opposed to that, a data scientist is somebody that can think about the problem and translate a problem presented by, let's say, a biologist into a specific technical description and an analysis plan. Now that plan starts from structuring the data, which is what we'll talk about today, but also being able to conceptually explore the data to understand what types of information are there, what kinds of variation need to be adjusted, how to prepare that data for analysis. We'll also talk about how such data could be used to ask specific questions. And in many cases, these specific questions are going to be based on availability of a certain number of data. So that data could include 3 samples, and that means that we can only test a small hypothesis, or it can include 3,000 samples, and that means that we will face this problem of big data. And so we'll talk about this trend of big data emerging in multiple domains, including oncology, neuroscience, agrotechnology, biotechnology, pharmaceutical companies use that data. And we'll talk about how to apply big data analysis tools for these large-scale data sets. And obviously, we'll talk about how to statistically use appropriate methods to analyze data, including machine learning that will allow us to explore data structures, that will allow us to understand similarity between many different types of data sources, and also use classification or predictive learning to apply machine learning for novel data that is being generated. And importantly, I think what we need to go back to is also how to explain what we have found in such analysis. So we'll talk a little bit about biological interpretation and how do we actually use all of these insights that we have found. What are some practical applications for those? So what this program will do is provide you with a complete roadmap for data science. It's going to introduce you to a lot of resources, and we do not expect you to complete all of them during the course of this program. But we hope that you will take out of this experience some understanding of how to find data sets, explore and visualize them, use them in research methods when you read a paper, how to interpret the research methods, and also gain insights and understanding about some practical skills. And so, as you can see, this is kind of like navigating a maze, because there's so many different directions to go. You can spend all of your time on just finding data sets, or you can find all of your time on just learning how to code. And we hope to combine elements of data science together to provide you with a roadmap. And we call this roadmap OmicsLogic. So that is the logic behind the analysis, the logic behind structuring your data into some format that you can ask questions and interpret the answers that you are getting. The program is going to be divided into two parts. The first part is going to deal with different omics data types, big data challenges, how to clean, load, and process data, how to understand what type of data and what kind of information is included in that data, and getting some hands-on experience with doing coding in R and Python, just to understand what do those languages offer us. It will also introduce you to a number of different example projects and some processing pipelines that you will probably not be able to conduct on your own, because they deal with a huge number of resources that need to be available at a certain time. So we'll leverage that big data infrastructure to try and run big data analysis. The second part will talk about statistical analysis, introduction to machine learning, and how to perform your basic analysis and evaluate that analysis in terms of accuracy and in terms of how generally applicable it is for other data sets. So we'll talk about supervised and unsupervised machine learning. We'll talk about model accuracy and validation, and we'll also talk about the difference between exploring, analyzing, and actually getting results with machine learning. Now, importantly, as I mentioned, this will be applied to a number of different project examples. For example, we'll talk about diagnostics and cancer biology. We'll talk about how to analyze single cell data and tissue composition and what we can learn from such data. And we'll also talk about the microbiome and infectious diseases. So we'll talk about how can we analyze specifically the data that was generated to represent species diversity or species richness, and how we can use such information to ask questions about different diseases or even non-clinical conditions like anxiety. So as I mentioned, an important part of all of these types of omics data is that they have to be processed. So essentially, when we talk about generating such digital data, we're talking about taking a sample and applying a specific technology that extracts some portion of that sample, of that biological material, to produce a digital data describing genomic variation, epigenomic regulation, transcriptomics, how genes are expressed, or even how proteins or metabolized are expressed. And so that processing of data is going to be a major limitation for anyone that wants to apply more advanced data science methods. And in fact, it is known that about 80% of a data scientist's work is transforming, cleaning, and preparing data for analysis. So we'll spend some time to talk about what are some of the considerations around using these big data analysis tools in a logical way, because ultimately what we need is a map that combines different methods together into what's called bioinformatic pipelines with explained methods. So we'll talk about what each one of these methods means, and we'll talk about how to prepare them into reproducible workflows that you can share and understand yourself, but also share with others what you have done. And we'll try to see how such processing allows us to compare different conditions, extracting only meaningful differentially expressed genes, how we can use multivariate analysis of big data when we have multiple features and multiple types of differences that we want to combine into a single analysis, how to also think about the application of we had found, if it's identification of protein targets, single cell deconvolution, or how do we map and represent complex relationships using our understanding of similarity between different types of organisms and genes. So at this point, let me share the poll results. Okay, and let's take a look briefly at what we have in this group. So it looks like for what country are you from? The majority here are from other countries. And so we also mentioned many of the countries that many of you put in the chat box. So thank you for that. What is your level of education? Looks like the majority of the people here are with a master's degree, and the second largest category is with a PhD. For the area of interest, the majority of you are interested in bioinformatics, and the second major group is in data science. And then we have what are you expecting to get from this program? And the majority of you are interested to learn about data science or bioinformatics. Now, the reason I am showing you these poll results is because the poll results are structured, right? I asked questions and I got answers in the exact format that I was asking. The answers that you wrote in the chat, as we were reading them, it's very hard to get a sense of how many are, for example, from Pakistan or from Finland or from Argentina or from Korea. I have to go back and I have to analyze what I have in the chat box, also assuming that many of you put things in variation. Somebody used a capital letter Pakistan, somebody used a lower letter Pakistan, right? So there's some processing that has to be done with this unstructured data. So that's why, in the context of this program, let's think of this poll as an experiment. We need to understand the experiment design. So when I asked you these questions, I had something in mind. I wanted to understand some specific question. Now, another person can look at the data that I have collected, and maybe somebody else's data that they collected, and they will look at how this data is organized and what the numbers show, and they will try to make inference. They will try to understand what that means in general about some population of people. So when we think about how these data sets are generated, we have to keep that in mind. We have to see that what we are dealing with is data that was produced by design. And so when that data was collected, we'll talk about how it is collected, what are the different mechanisms of generating the actual digital data that we deal with. Then it has to be cleaned. For example, if I look at the category called other, do I keep it? Do I try to structure it? Do I eliminate it from my data set? I need to create tidy data, or data that is ready for analysis. And I also need to understand how this data represents the experimental conditions or groups of samples that I have. Okay, so we'll talk about exploratory data analysis, where important things are visualization and data mining, or visually trying to look at my data and seeing what's in it, or using some additional methods that help me find more nuanced patterns that I might not be able to spot easily with my eyes, but nevertheless, they represent some trend, some pattern, and not noise. They're not random. Finally, we'll talk about data analysis. So an important consideration in data analysis is selecting out of many statistical tests and statistical models, how do I actually find the right one, knowing what my data represents and knowing what kind of result I am trying to get out of my data analysis process, and how do I share the results of the model by providing summary statistics and knowing what these summary statistical measurements mean. Finally, we'll talk about inference. How do we take what we have learned and interpret it to represent a larger trend that we would like to generalize? So in many cases, we do not have the ability to sample all of the people in the population or all of the samples that represent some kind of a condition. We take a sample, and that sample will be used to generalize our understanding, and that has to do with how robust our analysis is based on collected data, selected model, and the results of statistical analysis. Okay, so at this point, I want to just pause and make sure that we address any questions that you might have so far. So I saw a couple of questions here, and let me briefly address them. So some of the questions were about registration. I hope that we answered those. Some of you answered about asked about how all of these resources are going to be utilized. I hope that we are starting to answer this, and I'll conclude our session today with some more information about it. Another question about this paid course or the free webinar. So again, the way that this will work is you will join sessions like today. We will have some assignments that we will do together. We'll get to that in just a second. You will see what that looks like. And so if you want to participate in the full program, you have to benefit from both. But if you don't have the time and you don't want to spend the time to come in at something, at a session like this, you can also join the much more accessible and affordable asynchronous courses, and Beepsa will share the links. So once you register, you will see what those include. And I think that will also explain how is this course different from Coursera and edX. So a lot of the focus here will be on biomedical application of data science, and we will do a lot of practical analysis of data and source kind of evaluation of different sources of data so that you can actually ask biologically relevant research questions of that data in the context of the program. So it's a lot more personalized and a lot more practical, I would say, than, let's say, Coursera or something like that. Okay, can the results obtained be used for publishing? Publishing is not an easy challenge. I think that many of us coming from research background, you will know that a typical research article takes maybe between 6 months to a year, maybe even more. There's a reason why PhDs take a long time. After you've gained that experience, I think it becomes a little bit easier, but no one will likely say that publishing is easy. So we will be able to go through the process of exploring different sources of data, using big data analysis tools, data science in R and Python, and biological interpretation to, in the end, get some results. Those results could eventually be turned into a publication, but it's likely going to be taking longer than just the 1-month program that we're talking about now. We do have a research fellowship program that is more for those people that already have the basic skills, but would like to work on a research project with a group of other research fellows that we have and with our mentors. So thank you for your questions. Beepsa will add some more answers in the chat and some links. And for us to get most out of this session, let's go on and get to some practical examples of what we will do in the program. Okay, so if you do have any other questions, you can continue posting them in the chat box. And Beepsa, if you could please share the links again for those that have joined recently of how to register, how to get access to the educational portal, how to get access to the coding, and to look at the platform that we will use. Okay, so as I mentioned, the first problem is that we will be dealing with unstructured data. As a general trend, about 80% of the data that is being generated today is unstructured. That means that there was no model for this data. This data is not like a survey that had specific questions. It wasn't in a structured format. And if you want to think about how the data in biomedical research is unstructured, you can think of a vial of blood. So as you can see right here, right? So somebody collected blood, and then that blood had to be turned into genomic sequences. So the way that works is that there's a library to extract specific types of DNA portions out of the blood. And so there's a whole biological process in the lab that's being done for that. And then those pieces of DNA, those fragments of DNA that you can see right here, are shattered and sequenced. And when they are sequenced, we are essentially dealing with a lot of short DNA sequences. And one of the major challenges is to work with that unstructured data and structure it, right? And so as I mentioned, about 80% of your work as a data scientist or a data engineer is to be able to understand how the data was generated and turn it into a structured format. And that includes accessing and loading the data, cleaning it, understanding what it means, and then preparing that data for analysis. So we will look at how to apply different types of processing pipelines to make this into a structured data set. Okay, and Lydia is asking here, I have RNA-seq data, can I work on my research in this course? Yes. So if you already have some research data that you would like to analyze and make sense of, learning about the different principles of analysis, this is exactly the type of a program that you would want to join. Now, the other type of data is structured data. And structured data, as you can see right here, includes a organization structure, right? So this means that we have here a table of expression. In this table of expression, we have different types of genes represented by a gene ID and different types of cell lines that also have a different class. And inside this table of expression, we see the levels of expression, right? So just a question for all of you. How many genes are there in the human genome, approximately? Again, for those of you that don't have a data set, we will actually show you how to find some data set. All right, excellent. So we have here some answers. Sharif, Perna, Kabia, okay, 25,000, 30,000, 32,000, 20,000. All right, excellent. So yes, you know your stuff. So it is approximately 25,000 genes. However, as you can see from the answers that people are providing here, there's actually a variation. And the reason for that is because the human genome that was sequenced almost 60, 70 years ago is still being studied for what do we mean when we say a gene, right? And so a gene sequence is a fluid concept that continues to be expanded. And so the number of genes that are being discovered is also growing. And so I think that's one of the things that we need to remember is that we will deal with data that continuously is updated in terms of our biological understanding, in terms of the number of elements that we're going to be looking at. And now if you think about each gene representing multiple transcripts or multiple types of the ways the exons are spliced together, you will see that we can easily get to anywhere between 20,000 or maybe even 10,000 in a cell line that is very specialized, all the way to about 60,000 for some cases if we include different transcripts. So for each sample, we're dealing with a very large number of data points or features that we're going to be dealing with. Now, as we structure the data, we need to think of several different ways that we can organize this data in a particular structure. And in the case of our language, which is something that we will try to use today, there are several different ways that we can store this information. For example, we can use what's called a vector. A vector is a one-dimensional array that is used to store a collection of data in the same mode. For example, you can have a number of, a list of numbers, okay? And that could be a vector. There are numeric vectors, there are complex vectors, there are logical vectors, and there are character vectors or text strings, so different modes for vectors. There are also matrices. You can see here matrix. A matrix is like a table, but it is only used to store data of the same mode. So if you have all numbers and they're just the cells over here, so just the numbers here could be turned into a matrix. So you have to remove all of these strings right here, and that could be turned in and stored in a matrix. Multiple matrices or matrices together could form an array. And so an array could be multiple tables like this. You can also have factors. So these could be categorical variables designed to group the components of another vector with the same size. So if you look at this table right here, who can tell me where is a factor? Where could I create a factor for this matrix right here? Okay, excellent. Thank you, Vaishu. The class row right here if I take these, I can turn them into factors, because essentially you can see that they are repeating and they represent the type of sample that I'm dealing with. Okay, and so this could be factors, these could be names of my samples, these could be names of my features, and this is a matrix. Altogether, this could be a data frame, or essentially a table. Okay, so let's remember these definitions as we will go in today's practical part of today's session. Okay, and for Prerna, she was asking to repeat about factors. So again, factors are, you can see here, right, in this data frame or in this table, we have names of genes, we have names of our samples, but the samples fall into specific categories. In this case, we just see here one category, luminal cells. And so the factors, let's say I have condition and control, and I have 3 samples in condition, 3 samples in control. If I want to ask what factors do I have in my table, I would say 2 factors, condition and control. How many of each one? 3 and 3. Okay, so those are factors. If you think about your experiment design, when you're designing your experiment, you want to compare something, you design those by experiment factors. Okay. Now, the other important part of working with this structured data, once it is prepared, you want to analyze the data for how it represents what you know about the data and how that data is generated. Okay, there are 2 considerations here. The first one, let's talk about a biological experiment. As we said, we have 3 data sets or 3 samples in our condition and 3 samples in our control. If I take 2 samples from the condition or 2 samples from the control, so 2 samples of the same kind, I need to know that I can accurately represent that condition with those samples. And the way to compare them is I can use a model to assume that they are correlated. So if they are correlated, if the gene expression values are correlated, I know that I can use them to represent that condition. If they are not correlated, I know that there's some other pattern and I might be forcing my model onto the data. The other part of it is that this type of a comparison is not going to provide me with the right kind of values if it does not follow some assumptions about the data. And so another consideration here is what's called the distribution of data, or how many of each one of my numbers fall into a bin based on some kind of a distribution of the full data set. Okay, and so we'll talk a lot about these, so don't be overwhelmed right now with all these considerations. If this is completely new to you, this is something that we'll deal with in detail throughout the program. So we first structure the data, we have to put it in a right object, and once we have that object, we also need to understand the properties of that object that include some kind of exploration of the data characteristics. And so here we'll talk about some of the ways to prepare the data for analysis. A lot of that has to do with our understanding of the biology behind what the data was created for and how. And so a lot of the methods that you will see will include considerations around thresholds and around an understanding of what's the bulk or the noise maybe, and what is the specific signal, or what are we actually trying to compare. So when we talk about biological data, it is known that biological data does not follow a normal distribution in a raw format. It also represents a lot of noise that biologically is meaningful, but also can introduce a lot of noise into our comparative model or our contrasts. But also, we know that there are specific technical things that we need to understand about the process of generating the digital data. And so we'll talk about how to address those as well. Finally, we'll talk about how to present our analysis in a meaningful way. So we'll talk about things like heat maps, scatter plots, principal component analysis, etc. And we'll talk about what is the meaning of what we are seeing, how do we interpret what we are seeing, and how do we make sure that what we are presenting, if we want to share our results, looks meaningful to somebody that is going to be looking at it from a technical perspective, that it's correct, but also from a biological perspective, that it immediately is clear what you're trying to say. Finally, we'll talk about data mining and classification. And so here we have a lot of different examples of these methods. And I want to ask you a question. Who can point out one example here of classification? So just one example of a classification method. Again, you can type your answers into the chat box. So is there a classification method? Okay. Okay, so some people are mentioning clustering, k-means clustering, decision trees, hierarchical clustering, okay, clustering, all right. So again, classification is not clustering. Clustering helps us understand groups of samples in our data, and classification is used to predict a class based on learning. So we'll talk about this in greater detail, but I'm glad to see that there are some, some people know maybe more about this than others. But this is an important way to think about our different types of methods. And when we think about these methods, we need to understand what are some of the considerations. For example, why do we not use linear discriminatory analysis for complex data? Or what is the difference between random forest and decision trees? Or how can we think about hierarchical clustering and decision trees? How are they similar and how are they not similar, right? So we'll talk about all of these different aspects of machine learning, data mining, classification, and we'll think about how to also combine these methods together so that we can use them effectively. Now, in these sessions, we'll not only go through slides and talk about different hypotheticals, right? What we'll do is we'll actually go through very practical examples. So let's go and do a little example together. So if you could go with me to code.omicslogic.com. Hopefully all of you have already signed up there. And this is going to be a free course, so you don't have to worry about paying anything. So this is a completely free resource. What we'll do is we'll do a very quick example of how to load data in R and understand what is the data set that we are dealing with. Okay, so if you go to code.omicslogic.com, please let me know once you are logged in. Okay, so it is code.omicslogic.com. Okay, so once you are here and you're logged in, you can see that I'm logged in. You go to courses. Okay, so please let me know once you're inside and you see the courses. What we will be going through in this session is an example over here for Introduction to Data Science. And we'll just do one quick example from R. Okay, so please come with me to this first lesson, how to load data and check for variable data. And what you should see on your screen is something like this. So please put number 1 in the chat box once you see what I see here on the screen. All right, thank you, Vaishu, Sharif. All right, great. All right, so we have about 70 people here on the call. So if you want to put number 1, I'll give it a couple of minutes. And in the meantime, I'll tell you just a little bit of an introduction to data types and data structures in R, just to kind of refresh everybody's memory. So even if you have completed, we'll do something a little bit different from exactly what this lesson does. So please stay with us, Najiha. Okay, so to make the most out of using R language, you need a strong understanding of the basic data types and data structures and how to operate them. So what you can do with them, how to use them effectively, etc. Data structures are very important to understand because these are the objects that you will manipulate on a daily basis in R. Dealing with object conversions is one of the most common sources of frustration for beginners. In fact, I would say one of the major challenges that many people face is how do you actually get a data set into something that you can work with? So a lot of tutorials start with, let's do 1 + 1 or something like this, right? But here we want to deal with biomedical data. We need to load and understand what's in it. So R has 6 basic data types: character, numeric, which could be real or decimal, integer, logical, or complex. So elements of these data types may be combined to form data structures, for example, atomic vectors. So when we call a vector atomic, we mean that the vector only holds data of a single data type. So here are some examples of atomic character vectors: numeric vectors, integer vectors, etc. A character could be A. Numeric could be 2 and 15.5. Integer could be 2L. Okay, the L tells R to store this as an integer. Logical, which is true or false, and complex, for example, 1 + 4i. Okay, so those are vectors. And also, R has many functions to examine features of vectors and other objects. For example, you can use the functions class, type of, length, and attributes. Okay, so let's think about how we can learn about this in this example. Okay, so you will understand the problem. You can see that we can import data directly here. And let's look at what is the basic code. So the basic code here is to load a table where you need to include a path for that table. And you need to also specify some things about the table. So you need to know what the table is like. For example, it could be a tab separated table. You would use a /t. It could be a CSV. So it could be a comma separated. So you would include a comma here. And it could either include or not include a header, right? So as we saw in our example here, we can have a table, and that table can contain some information in this first line. That's called the header. So we can specify that right here. And obviously then we can print what we find in our data. So let's go to next, and we'll see a little code block that we can use together. Okay, so here you'll see the first code block. What it includes is a function called readTable. And because we're going to use a table, we need to put here a link to a txt file. So if you right-click right here on this link, you can use copy link and then replace the path. So you can see we're using here biologically height dataset. We'll keep header as true, separator as tab delimited, and you can just click on run. So because we are printing this object, it is going to tell us what this object contains. And you can see that this is a table. There's an ID, specimen 1, 2, 3, and 4. There's the type of specimen, flower, tree, and bird. And there are some numbers in here. Okay? Now, let's use the function class. Okay, so what is the class of what we have loaded? It is a table, right? So it's a data frame. Okay, so now we have different types of attributes inside this data frame. And so here we can use type of and say again df and do run. And you can see that the first one is list, right? So because it's going to look right here for a list. Okay, so and so forth, right? So you can actually start playing around with this data set because you have loaded into a data frame. Okay, so this is just a very brief example. You can also practice on how to do this with Excel or a CSV file and some other biological data sets. The key point here is to understand how to load a data set and also how to explore what type of data have you loaded. Any questions about this? All right, great. So I hope that this was clear, right? We have some data that we can look, and additional types of analyses in R and in Python will go through a lot of the same methods. Header true, okay, so just to explain again, what is header equals true? Yeah, so this is a free resource. There's a lot of free coding lessons here. You can go ahead and explore them. Some of them have also paid lessons, but if you join the program, you will see how it's all combined. And it works best when somebody's guiding you through this, because not everything could be explained easily in an online tutorial, okay? Yes, the path should be in quotes. What is the header? The header is the first line. The first line is not the data itself. You're just telling you're just specifying that this is going to be used as names of samples and are not to be included in the actual analysis. All right. Now, as I mentioned, there are a lot more courses like this that we will go through in this whole tutorial. And the objective of this program is to try and piece together this puzzle of different types of projects, different types of data types and sources, and different types of practical analysis techniques to help guide you from where you are at a very beginner level or you are more advanced to actually make progress and gain experience with analysis of data. Okay, and Rudina, if you are getting an error, cannot open the connection, maybe it's because too many people are doing this at the same time, just reload the page. Okay, so combined together, we will use these three different resources. Educational training materials, which you can get access to online without the whole session, like we're doing it interactively. You can gain access to a number of different educational courses. You will have a platform for big data analysis. You will also have coding exercises, and together you will really gain a perspective of how data science can help you explore different type of data that are genomic, transcriptomic, metagenomic, metabolomic, proteomic, etc., to explore variation and make biological interpretation. Now, as I mentioned, an important part of that is being able to reproduce workflows that others have done. So you will see how we will go through the different publications that explain this data and explain the method of analysis and turn that into a tutorial for you. So we will look at examples of sequence alignment, RNA sequencing, evolutionary analysis, machine learning, variant calling, and structure analysis, all for you to make the most out of the different examples so that you can use this in the context of what you are interested in. Now, what we saw today is just an example of an online solution that we will use to make sure that everybody's on the same page, but we will also introduce you to RStudio and Jupyter Notebooks in Python so that you can take what you learn and use it on your own computer. So for those of you that are new to this completely, you can actually watch this little video that will tell you how to install RStudio on your computer. And so in the first few lessons, we're going to be using R a little bit more, but as we go more into the machine learning part of it, we will also start using Python, okay? So here you will see how to, in this video that I shared, you will see a basic explanation of how to install RStudio on your computer, and then also set up some basic things like your working directory or installing different packages. Now, the benefit of what we are going to be doing in this program is that we are going to be combining all of these different elements together to help you make the most out of different types of tools and also understand them in the context of specific examples. So all the data sets, all the different packages and functions that you will need are pre-installed on this online solution. Ultimately, you will gain experience on visualization, statistical analysis, and ways to interpret the data that you have. And so here we will see examples like principal component analysis, visualization, and other things. And so the main objective here, as you can see on the side, is to understand the problem, learn the code, learn to modify the outputs, and then modify settings and apply to the data. And hopefully by that time, we will do a session tomorrow, or not tomorrow, in our next session, where you will see how to find data sets. So if you have a data set, we'll talk about processing. If you don't have a data set, we'll show you how to find data sets that you can work with. And the next session is going to be on January 14th. Thank you, Deepsa. So the project part of this program is very important, and we hope that all of you will be able to get through some of the basic things. You can see here we track the activity of all of the users so that you can get a sense of where you are. And we track these three different things: utilization of the platform, completion of courses, and how you complete coding challenges. And you will first get an introduction to some basics, and then you'll be able to apply what you have learned to a project. So we hope that you will come out of this program with a project of your own. To get you started, we also have a number of project examples that we have prepared following some publicly available data sets that you can find on NCBI. For example, one of these will be modeling cancer precision medicine, where we take a publication on different breast cancer cell lines and look at how to analyze such data to understand the differences between these cell lines. And in each one of these data sets, we will go through the publication and we look at some keywords of what you will actually learn. Here you can see mentioned in yellow the conceptual ideas like molecular profiles or molecular pathobiology, and also the specific methods like support vector machines, random forest, mutation, gene and isoform expression, etc. So hopefully after this session or after this program, you will be able to easily read through the publication, understand all of the terminology, but also learn how to interpret these visual abstracts so that you can take this on with you to your own independent project. Now, we've prepared a number of different examples. They range from deadly mutations in Ebola virus to the origins and pathogenesis of the novel coronavirus, infection and treatment of tuberculosis, insect cell lines contamination and production of biologics. So we have a lot of different examples that you can start with to get you ahead so that you don't have to go from very basic kind of trying to understand why the data was generated, etc. So at the end, you will get a certificate of completion for the full program, and you'll be able to also get individual certificates for courses. And again, we have thousands of participants that have joined from many different backgrounds, including people that have postdoc positions and research positions at different clinically relevant research positions that you can actually apply this in the context of biomedical research. And many of the participants actually that have joined and completed these previous programs have been able to compare this program to some other alternatives on edX and Coursera. And so we have a blog post by one of our participants who works as a bioinformatician at a genetic testing company called Atlas, how this is different from some of the other programs out there. I know that some of you have asked, and I thought that maybe it would be useful. So if you could share the link to that blog post as well. So again, what we will study in the first part of this program is how to deal with data, learn how to load it, learn how to manipulate it, and explore it. In the second part of the program, we will learn about statistical analysis, machine learning, and how to do this in the context of specific projects. After the basic training, you will be introduced to projects and data sets that you can find online on your own, or use your own data sets to do the analysis as you have learned in the program. Okay, so that was the end of this presentation. Thank you again for bearing with me. I know it was a little bit longer than expected, but at this point, if any of you have any questions, I would be happy to answer those. We also have here with us Deepsa Biswas, who's our community manager. And if you have any questions about registration, payment, access to different resources, updating your profile, etc., please reach out to her. It is marketing@pine.bio. We have our project mentor, Dr. Mohit Mazumdar, who is going to be helping those of you that decide to go on and work on an independent research project. So you can reach him at mohit@pine.bio. And we will also have Dr. Harpreet Kaur starting from our next session. Her email is support@pine.bio. If you have any technical questions about bioinformatics, R packages, Python, loading your data, getting a specific type of visualization or an error resolved, please email her as well.
Ilya, I do have a few questions here to address.
Okay.
One that I would want to address is the question whether, so I got this question from a lot of participants attending today that they may not be able to make time for the sessions which start at 9 a.m. CST. So in case you are not able to make time at 9 a.m. CST due to other obligations which you may have, we always record all of our sessions. So if you're intending to attend the program, it is going to be a 1 month program which is spaced out, and I will again be sending the schedule for the program with everyone. So we will record every day's session, so you can watch that video later, and that video will remain with you because we upload it to a private playlist on YouTube and share it with only the participants who are attending the program. So you will have exclusive access to that throughout the range of this program and beyond that. And in case you also need mentor support, as I already mentioned, you can actually ask for mentor support who will be able to help you with any questions that you have during the session. And here is a question, Ilya, which I think we should answer, like, is multi-omics used exclusively for research purposes, or is it able to be used commercially amongst the medical community?
Yeah, so I would just mention about that the platform that we're going to be using here is a platform that is used by many different medical researchers. As you know, research in the omics era is becoming more and more commercially viable. So analyzing data sets from transcriptomic, genomic, epigenomic regulation data together about the same phenomena is extremely powerful because you have the additional validation of an independent source of data about the same probably messy and noisy situation. So multi-omics data sets are generated in a biomedical research, translational research context. I would say that it is very rarely generated in an actual clinical setting because clinical refers to making a specific decision about it or maybe having a payment code associated with that analysis, and it's not there yet. But it's definitely moving in that direction. Now, in terms of using the multi-omics analysis platform, we have a lot of regulatory oversight over the platform hosting and data management. So if you are interested in analyzing proprietary data of clinical significance, you can get a research account which will provide you with that security as well.
Yes,
Okay,
and
so
anybody
yeah.
has any other questions, you can always reach out to me, marketing@pine.bio. And I just wanted to let everybody know that the discount code is supposed to end by today. So there is a 20% discount that we had been offering. So you can check your emails for that code if you are interested. And if you're joining, if you have any other questions, please reach out to me, marketing@pine.bio, and I will be able to assist you with any queries, even related to your research fellowship program or the research license for the T-Bioinfo server, or any other questions that you may have regarding projects, as well as the data science program. And thank you again for joining, and thank you, Ilya, for taking us through this lecture today. So we hope to see you in the next session.
Okay, great. Thank you, everyone. Looking forward to our next session, January 14th.